Hi, so chapter 12 goes over the nervous system physiology, and let's begin by going over a basic model of how it receives and coordinates information. The neuron is the functional unit of the nervous system, and it's how it passes information in the form of electricity from one place to another. So it uses this basic model here, this funneling looking system, and you're going to see this in lab, probably already have, and you'll see it again in A and P2. So it receives information through a neuron, and that neuron is called a sensory neuron, or afferent with an A, and then it sends that information to the central nervous system that coordinates that information, processes it. And remember, the central nervous system includes the brain and the spinal cord. It integrates that information and then it sends it out of the central nervous system to the peripheral nervous system through a neuron called motor or efferent with an E. And it coordinates this action from an effector. And the effector could be a muscle, it could be a heart cell, or it could be another neuron. So we'll see this basic model throughout the nervous system, and even your endocrine system uses something similar to this. So we'll see this here, and we're going to see it again in A and P2. So whenever you think of cells of the nervous system, perhaps the very first thing you think of are neurons, which is the functional unit, but it only makes up 10% of all the cells of the nervous system. So there was this guy named Camilla Golgi, and he dropped this brain in silver nitrate, and it turned all these different colors. And then he realized, oh my gosh, that only 10% of all the cells within the brain are actually neurons. And the other 90% is made up of these cells: microglia, oligodendrocytes, astrocytes, and ependymal cells. So in this picture, we have, of course, a neuron, but we also have all these other cells. We have microglia, which function as immune cells, like macrophages that eat debris. Immune cells are like the white blood cells that have moved out of the blood into the tissue. In this instance, the nervous system tissue, and they're cleaning up dead tissue, damaged tissue, bacteria, whatever it may be. So it's like the immune system of the nervous system. Oligodendrocytes, which make myelin sheath. So here is an oligodendrocyte, and they make the myelin sheath particularly for the central nervous system. Okay. Astrocytes. Here is an astrocyte. They play a role in the blood-brain barrier. So look here, they have little hands that are connected to the capillary and also to the neuron. So you don't have a blood supply directly into the nervous system tissue. There's a barrier there, and that barrier is played by the astrocyte. So it's like another stop. It says, hey, I'm going to decide what gets in and what gets out. One of the things that does get in is actually alcohol. Okay, another cell is the ependymal cell, and these are found here. They help produce and circulate the cerebral spinal fluid, the CSF, the cerebral spinal fluid. So in lab, you look at a model, this little gray model of the ventricles found within the brain, and if you turn it over, there's a little pink area underneath the ventricles, and I think you'll call it the choroid plexus. Now, this is where those ependymal cells are located, and they produce the CSF. So make sure that you know these cells and what they do. So put you a star by that, because I will definitely ask you what they do on the exam. Here we have a picture of a general picture of a neuron with its different parts labeled. Make sure that you're familiar with the different parts of a neuron. So here we have dendrites, the cell body, axon hillock, the axon itself, and then the end, the axon terminal or synaptic knob. The other name for that is bouton, which is actually French for button, which is kind of funny. So dendrites are actually defined by the direction, and what they mean by that is the direction in which they receive info. So dendrites take information towards the body, the cell body, or the soma. So if it is a dendrite, it's going to be taking information towards the cell body. Now, from there, it takes information down the axon towards the synaptic knob or the axon terminal, and then information is then going to pass across the space, which is referred to as the synapse. Make sure you know that. And then find its home on an effector. And remember, an effector could be a muscle cell, a heart cell, or even another neuron. Now, remember, the neuron is a cell. So within this space here is cytoplasm. And that means that we can move charged particles across that space within that cytoplasm. And that is actually how we create this action potential. Okay? Electricity is the moving of charged particles. They move down the neuron, and to do that, we need a difference of charge. In cells, the inside is generally more negative than the outside. Okay? And that's due mostly to the sodium found out here, because they have that positive charge. And when sodium moves into the neuron, that's basically electricity. Now, the movement is called an action potential, AP for action potential. The wave of charged particles moving down the neuron is an action potential, and we'll talk about this more. So how do we actually get it to move? Well, we need a difference in charge. Okay, so we have to create this difference in charge in order to get this wave. One other thing I wanted to tell you before we move on is that down the axon itself, it is an electrical message that is moving, because we're moving those charged particles. But once it gets to the synapse, in this space, an electrical message cannot pass. And so we have to have a chemical message. And that's where we get those neurotransmitters, NT for neurotransmitters. So that is then a chemical message. So down the axon, it is an electrical message, but across the synapse, that space, it is a chemical message in the form of what we call neurotransmitters, NT for neurotransmitters. Okay, so who do you think it is that creates this difference in charge? And what I mean by that is the negative inside versus the positive, more positive outside. And the answer to that question is the sodium-potassium pump. Remember that sodium has a positive charge, as well does potassium. But this pump moves these things in opposite direction and in different amounts, and it creates this difference in charge between the outside and the inside of the cell. So let's take a look at how it actually does that. So in its original conformation, the sodium-potassium pump, which is a protein, it's an active transporter pump, which means we're going to use ATP. The very first thing that happens is sodium, 3 sodiums bind, and that creates an ATP binding site. Okay, that creates that ATP binding site. Then, this should be a green box here, ATP binds, and then the whole thing will change shape. And now it is open to the extracellular fluid, so the outside of the cell. Okay. Now the pump is open to the outside. The sodium, with its positive charge, the sodium binding site changes and no longer fits the sodium. So our sodium, our 3 sodium, falls off. So how many sodium have we moved to the extracellular fluid, to the outside of the cell? 3. So 3 positive sodiums have moved out. Now what happens? A new potassium binding site has formed. And then potassium will bind to our sodium-potassium pump. A phosphate from our ATP falls off. And then our pump is going to return to its original conformation like it looked before at the very beginning. And when it does this, our potassium binding sites change again, and our potassiums fall off into the inside, into the intracellular fluid. So we add 2 positive potassiums in. And that is what makes the outside of the cell more positive than the inside. Okay, so make sure that you know these numbers, and it is the sodium-potassium pump that does this. Almost every single cell has a sodium-potassium pump. You're going to see this over and over and over again. And it is critical for establishing the ability to make electricity in our neurons. So understanding this now is very important. Make sure that you know this. Put you a big star by that. Okay? So this is normal. This is what we're going to see establishes resting membrane potential for our neurons. So if a membrane potential is just the difference in charge between the outside and inside, outside being the extracellular fluid and the inside being the intracellular fluid inside the cell, what all contributes to it? Well, we know sodium does, and potassium, thanks to our sodium-potassium pump. So we have a higher concentration outside of sodium than we do inside, and a higher concentration of potassium inside than we do outside. But also things like chlorine, which actually has a negative charge, and calcium. Those all have a higher concentration outside. But on the inside, we have a higher concentration of proteins. And in general, those have a negative charge. Remember, proteins make up your organelles, and you have tons of organelles within your cells. And in general, they have a negative charge. And when measuring the membrane potential, what we do is we set the outside to 0 and just test the difference, and that gives us the membrane potential. The reason we can set it to 0 is because we're only concerned about the difference, like subtracting from something. If you only care about what the difference is between one and another, so it doesn't matter where we start. And what it ends up being the inside is actually a negative 70 millivolts. And a volt is just the measure of potential energy. Okay? And again, inside here is much more negative. So from this, how do we get moving charged particles? Right? Because we want it to move down the axon. And that is in the form of an action potential. Or AP. Action potential is the rapid change in membrane potential and then back again. So for example, if we start at negative 70 and go to positive 30 and then back down again to negative 70, that would be considered an action potential. And actually, these scientists named Hodgkins and Huxley in like 1963, they took these giant squid axons that were millimeters thick, they were big, and observed these action potentials. And they did this by putting a voltmeter in the actual axon and then firing it and firing the neuron, and they were able to actually see this happening, this membrane potential to change, to go from negative to positive and then negative again. And this is how they described an action potential. They did this by observing the change in giant squid axons. So that's pretty cool. Alright, so make sure you know what an action potential is and the concentration that we find outside versus inside that contributes to what resting membrane potential is. So now let's go over the steps of actually getting the action potential to move. And to do that, we need to identify the different proteins or pumps or channels that are involved. So we know we have the sodium-potassium pump. And let's just review that with the sodium-potassium pump, sodium is moved out and potassium is moved in. Okay? Now, you also have channels that are embedded within the cell membrane. You have sodium channels, and you're going to also have potassium channels that are embedded within the cell membrane. And what you'll find is that they're going to move things the opposite way in which the sodium-potassium pump moves. So instead of moving sodium out, it's actually going to move sodium in. And the potassium channel, instead of moving potassium in, it's going to move potassium out. So maybe make you a little note here so that you don't get confused. The channels move it in the opposite direction of the sodium-potassium pump. And then also, we're going to have a stimulus that is going to start off the action potential. So before all of this happens, we're sitting at resting membrane potential, which is negative 70. In most cells, it is negative 70 millivolts. So we're cruising along at negative 70, and then we get a stimulus. Now, a stimulus could be anything from a light to filling a spider crawl on your arm. So it could be anything. And what this does is it causes a stimulus-gated sodium channel to open. So from number 1 to number 2, we're adding positive sodium charges. And what that does is causes our membrane potential to become more positive, from negative 70 to negative 55. Now, negative 55 happens to be a special number. That's what's called threshold. And if we reach threshold, then something else happens. And to reach threshold, that stimulus needs to be big enough. You have to actually I guess in words, feel it in order to reach threshold. So if we reach this threshold, negative 55, then another channel opens. And that happens to be a voltage gated sodium channel. And so from step number 2 to 3, we're adding tons of positive sodium charges. So here we're seeing sodium rush into the cell, right? Rush into the cell. And we become way positive. So if we reach that magical number, then our sodium channels will shut. Okay? This is time sensitive. So from 3 to 4, not a lot happens. There's no real change in charge. So during that time, our potassium voltage gated channels open. And which way does it go, do you think? Look at our little chart here. Potassium is going to go out. So instead of adding positive charges, now we're losing positive charges. Okay? And it will continue to do that. It actually drops below a resting membrane potential. So from 4 to 5, we're losing all of our positive charges. Potassium is leaving the cell. It drops below resting membrane potential, and eventually those potassium charges will close. So who is going to put us back up to resting membrane potential at negative 70? Who do you think reestablishes our resting membrane potential, which is what our cell is doing at homeostasis at normal? That is the sodium potassium pump. So I guarantee you I'm going to ask you a question about that, so you better know it. Okay? In all reality, from step 4 to 5, or even before that, we actually have potassium channels that open, I think from here to here, but they're super slow. So at threshold, at negative 55, our potassium channels actually open, but they're really slow. So they don't hinder our climb towards that positive 30. Only when our sodium channels shut do those potassium channels really open up wide and cause us to drop back down negative. So that's why they don't really cause a difference in that positive membrane potential. So anyways, make sure you know number 6 for sure. Okay, and know which way that charges are moving. So let's take a look at this chart. This is the same chart we just did. It just has a few other words on here that are important to know. So we have millivolts on the y-axis and time on the x-axis, which is the horizontal axis. So we know we have that stimulus that starts it all off and moves us away from resting membrane potential, which is negative 70. And then we have threshold at negative 55. And as we climb more positive, they term that as depolarization. Depolarization. And then when we move back down negative, that is repolarization. And during this time, it's called absolute refractory period. And make sure that you know these two words. Absolute refractory period is where you are physiologically incapable of firing another action potential. Okay, and I wrote my numbers here a little off. It's actually probably about, it's only probably about 2 seconds, I would say, or 2 milliseconds, sorry, in which you're incapable of firing another action potential. And what this is due to is those sodium channels They're time sensitive and they'll actually refuse to open back up. This keeps the action potential going in one direction instead of going backwards. Now, the other word is relative refractory period, and that is located here. So after we've repolarized and we actually drop below resting membrane potential, which is -70, if the stimulus is big enough, you can have another action potential during this time period. That's only if the stimulus is big enough. Okay? And then following that, we just call it the resting. You're in the resting state. Okay? So make sure that you know these two words and what they mean. Absolute refractory period, you cannot fire another action potential. Relative refractory period, only if the stimulus is big enough. Okay? Now, other landmarks on a neuron that are important to note are the myelin sheath. And what myelin sheath actually is a form of insulation. So some of your neurons have this myelin sheath in the central nervous system. Those are produced by the oligodendrocytes. And then in the peripheral nervous system, those are produced by Schwann cells. And we'll take a look at those in the following chapters. But what I want to note here is that in between your myelin sheath, you have what are called nodes, or nodes of Ranvier. You think you talked about those in lab. So what happens is that this whole thing is when we fire an action potential, and that has to happen at like here, 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 and then again, and again, and again, and again along the axon. Okay? But if you have the myelin sheath, it can go from node to node to node to node. So instead of having to happen along the entire length of the axon, it can happen at the node and continue its same charge, like hold the charge across the myelin sheath. It's insulated. And then it can do another action potential here, here, and here, and then hold it, and then again, and again, and again. And this is actually called saltatory conduction. It's allowing you to maintain the charge until you get to the next node. It helps you to move the electricity, the charge, down the axon quicker. So then we'll look at diseases that demyelinate or destroy the myelin on axons and what that does to the conduction of the action potential. So make sure you know what this means. I will definitely ask you about it. Saltatory conduction, allowing you to maintain the charge till you get to the next node. So far we've just talked about what a chemical synapse is, and we did this when we talked about muscles, but you can also have an electrical synapse. So reviewing what a chemical synapse is, remember at the end of the axon, it's the axon terminal, the synaptic knob, or the bouton, whatever you want to call it, the action potential, which is the charge, reaches the end, and then what that does is, number one, opens a voltage-gated calcium channel, and calcium will come in. It finds its receptor. Let's write that down. Calcium rushes in. Calcium binds to its receptor on a vesicle, our vesicle here, and that vesicle, remember, is holding a neurotransmitter. And once calcium binds to it, then that vesicle exocytoses the neurotransmitter out of the cell into this space. And remember, that space is called the synapse. So this is an example of a chemical synapse. And then the neurotransmitter here is going to find its receptor on an effector. This is just review. Okay? But we also have another type, and that's called an electrical synapse. And these are super important, especially in your heart. in your heart cells. So what this is for example, is we'll have one cell and then an adjacent cell, another cell. And the action potential will travel from one cell to another cell through what's called a gap junction. The action potential travels to the adjacent cell through this little gap junction, which is basically a tunnel. It's traveling through the cytoplasm from one cell to another cell, and then it can actually stimulate voltage-gated channels, okay, and causing other things to happen. So make sure you know the difference between an electrical synapse and a chemical synapse. So what does the neurotransmitter do after it's in the synapse? We know it finds its receptor, but what happens to the leftover neurotransmitter? So here we have our picture of our axon terminal. The neurotransmitter is in that space, and we also have our effector here with the receptors. Okay? So there are one of three things that can happen. It can reabsorb back into the presynaptic neuron. So that's a new word. The presynaptic neuron just means the neuron before the synapse. So that would be the presynaptic neuron. And that's what we're seeing happen here. Our neurotransmitter, the little blue dot, is being absorbed, reabsorbed back into the presynaptic neuron. Okay? Or it could be broken up with an enzyme. We've talked about one of the enzymes that breaks up a neurotransmitter. If you remember, what is the enzyme that breaks up acetylcholine? It was that real long word, acetylcholinesterase. I know that's a mouthful. So here we have our little Pac-Man enzyme breaking down a neurotransmitter. And then the third option is to have special types of cells called glial cells clean up that leftover neurotransmitter. So that would be here, and the type of glial cells that might do this are astrocytes or microglia. Remember, microglia are the ones that we consider the immune cells. Alright, so make sure that you know this. I know I asked you a question about what happens to the neurotransmitter that's left in the space, the synapse. So put you a star by that. Alright, so let's discuss neurotransmitters. We've talked about one before, acetylcholine. I abbreviate that as ACH. And they're often classified by the type of receptors that they use. Now, acetylcholine uses types of receptors called cholinergic receptors. And we've discussed one of those before, a nicotinic receptor. And what happens with nicotinic receptors is when acetylcholine binds, it actually opens a sodium channel and stimulates that action potential. So make sure that you know what nicotinic receptors do when acetylcholine binds. Another type of cholinergic receptor is a muscarinic. When acetylcholine binds to muscarinic receptors, it stimulates, instead of sodium, it stimulates a second messenger cascade within the cell, or within the effector. Okay, so make sure that you know the difference between those two for sure. Put your stars by those. It's also worth noting that our somatic motor neurons release, all somatic motor neurons release acetylcholine that bind to nicotinic receptors on skeletal muscle. So make sure that you know that for sure. And this is what we saw when we went over the muscle chapters. Okay. And here again we see that enzyme, acetylcholinesterase. Remember, if it ends in ACE, It is an enzyme. And this enzyme breaks down acetylcholine and helps to remove it from the synapse. Make sure that you know that. You can be sure that I'm going to ask you about that. Okay. Another neurotransmitter that is common is norepinephrine, and the receptors that it uses is the adrenergic receptors. So norepinephrine is an example of what we call a neurohormone. And what this word means is that it's one, a neurotransmitter, but also a hormone. And to be a hormone, that means that it's dumped directly into the blood. So it has a dual function. It can function as a neurotransmitter, but also as a hormone. So they call it a neurohormone. Now, talking about its receptors, they call them adrenergic receptors, and there's basically two types, the alpha and beta receptors. So that is all that I'll ask for you to know about their receptors for now. We'll go into them in more detail in A&P2. So know that norepinephrine uses adrenergic receptors. Other common neurotransmitters are serotonin. You've probably heard of serotonin before. Associated with mood, appetite, sleep, memory, perceptions of reality. Low levels are associated with risk-taking. It can actually inhibit pain in the spinal cord. Drugs like Prozac, which are termed SSRIs, and that stands for selective serotonin reuptake inhibitors. They block the receptors that would want to reabsorb serotonin. So here we have our axon terminal, and let's imagine that here is our serotonin in the synapse space, and it's trying to get back up into the axon. And Prozac comes along and blocks those receptors that would want to do that. So that would be how Prozac works. And in turn, it leaves the neurotransmitter, serotonin, in the space. And by doing so, it leaves it to find its effector for a longer period of time. Another common neurotransmitter you've probably heard of is dopamine. Dopamine is the reward and pleasure neurotransmitter involved in addiction, involved in motor function. Low levels are actually seen in Parkinson's. That's the one that's associated with tremors. We'll talk about this when we talk about drugs that affect addiction. I would suggest knowing what these neurotransmitters like serotonin and dopamine are associated with. So put your stars by that. So lastly, let's talk about methamphetamine, and that's a legal drug that acts a lot like dopamine. But before we do that, I want to talk about how they classify drugs, either as agonist or antagonist. So if it's an agonist, that means it's going to increase the effect of the neurotransmitter. It will look and act like the neurotransmitter. It will increase the neurotransmitter release into the synapse, and it can actually block neurotransmitter enzymes. So if it is an antagonist, which means it's going to decrease the effect of the neurotransmitter, it can block things like sodium-potassium pumps. It can block channels. It can also close channels, inhibiting the effect of the neurotransmitter. So then a drug like methamphetamine, what it does is it increases the release of dopamine into the synapse. It blocks transporters that remove dopamine from the synapse, and it can cause neurons to fire more. But eventually what happens is your body decreases the receptors for dopamine, or that much dopamine-like stuff eventually doesn't have the same effect. And this is what leads, or this is how addiction actually occurs. And then what happens is you have to keep taking more and more of that drug in order to achieve that same effect from that initial dose. So looking at these two types of classifications, what do you think methamphetamine would be? Either an agonist or an antagonist? So based on these things right here, I would say it is an agonist, since it increases dopamine into the synapse and blocks its transporters that remove dopamine. Okay, so that's it for chapter 12. Happy studying!